Welcome to the debate. Imagine an antique pocket watch.
Right, with all those tiny gears?
Exactly. Hundreds of interlocking gears, microscopic ruby bearings, delicately coiled springs. It's all ticking in absolute mathematical perfection.
Yeah, and if you shave down even a fraction of a millimeter on one gear, the whole thing just stops dead.
It fails. It's a marvel of uncompromising order. But, what happens when you take that perfectly ordered watch and subject it to a changing environment?
Well, I mean, if you drop it into a boiling geyser or bury it in a glacier, it freezes, it shatters.
Exactly. Because its perfection is completely inflexible. It only works under the exact conditions it was designed for.
But then you look at biology. You look at living organisms that survive in boiling thermal vents or in the freezing vacuum of space.
Right. Life is clearly not a fragile pocket watch. Which leads us to a fundamental mystery, really. How did life survive billions of years on a hostile, ever-changing rock?
And that brings us to the core of our discussion today, drawing from fundamental texts on genetics, cell division, and heredity. When examining the foundation of life, we're asking a specific question. Which principle is the true driving force of biological success? Is it the strict, predictable order of genetic replication, or the chaotic, unpredictable nature of genetic variation?
Well, I will argue that life's staggering success across billions of years is defined by strict adherence to genetic rules, precise replication, and predictable inheritance. The stability of life relies on an uncompromising order.
And I will argue that life's true engine is complexity, the breaking of those very rules, and the continuous generation of variation. Without the chaos of deviation, we would have gone extinct long ago.
So let's start by looking at the very manual of life itself. Biological stability relies on an uncompromising set of rules. DNA acts as a highly structured instruction manual, a double helix composed of precise base pairs.
Sure, the ultimate blueprint.
Exactly. And when we look at how life sustains itself day to day, we have to look at mitosis.
The classic cellular division.
Right. Mitosis is the body's perfect copy and paste function. If we look at the mechanism, it's astonishingly mechanical. The cell duplicates its DNA, tiny cellular cables called spindle fibers attach to the chromosomes, and they physically pull identical sets of DNA to opposite sides of the cell before it splits.
Making clones, basically.
Yes, it ensures that when a cell divides, it creates two identical daughter cells, maintaining the exact 46 chromosomes necessary for human growth and repair.
Look, I don't disagree that the machinery is essential. Of course, an organism needs to heal a cut with identical skin cells.
But it's way more than just a Band-Aid. Think about the sheer scale of precision required to maintain a human body, which is composed of roughly 37 trillion cells. Every single one of those cells, with very few exceptions, contains the exact same genetic code.
It's a massive number.
It is 46 chromosomes arranged in 23 pairs. If DNA is the source code of life, mitosis is the essential save and backup feature, preventing a systemic crash. I see why you value variation, but without this rigid 46 chromosome baseline, variation is simply destructive mutation. The organism must first survive the day before it can worry about adapting to the millennium.
I come at it from a different way. I acknowledge the necessity of mitosis, but maintenance is not the driving force of success. Maintenance just keeps the lights on.
Well, keeping the lights on is pretty important.
It's static, though. If you want to talk about the millennium, you have to talk about meiosis. Meiosis is the true driver of the future.
Just because it halves the chromosome count for reproduction?
Not just that. Let's look at how meiosis actually works. Instead of just creating a clone, the cell goes through 2 rounds of division in the reproductive organs.
Right.
Before it divides, it deliberately scrambles the genetic material through a process called crossing over. This is where homologous chromosomes physically align and swap pieces of DNA.
Wait, let me stop you there just to clarify the mechanics.
Go ahead.
When you say homologous chromosomes, swapping pieces, You mean the chromosome I got from my mother and the identical type chromosome I got from my father literally cross over each other, break and trade chunks of DNA?
Exactly. It's molecular origami. They physically snap and reconnect, shuffling the genes. Then, the cell reduces the chromosome count from 46 to 23. It creates 4 completely unique gametes, or reproductive cells.
Why call that the driver?
Because if we only relied on the perfect replication of mitosis, a single new pathogen, like a single virus perfectly tuned to the lock of that exact genetic code, would wipe out the entire species in 1 generation. By creating completely unique offspring, meiosis provides the raw material for adaptation. It ensures the species is a moving target.
I see your point about disease resistance, but calling it chaotic variation diminishes the incredible order governing even that reproductive division.
Order?
Yes, the reduction to 23 chromosomes in meiosis isn't random destruction. It is a highly orchestrated mathematical necessity. It happens so that when 2 gametes fuse, the resulting organism returns to the perfect, stable baseline of 46 chromosomes. The system demands a return to order.
It returns to the number 46, sure, but the sequence within those 46 is entirely new.
Expressed in Mendelian inheritance. Anyone listening who drew those little 4-box grids in high school biology, the Punnett squares, knows exactly what I'm talking about.
Ah, the classic pea plants.
The transmission of traits isn't a free-for-all. Those Punnett squares are a lovely teaching tool, but they paint a highly sanitized picture of heredity.
I'm not convinced by that line of reasoning because they paint an accurate picture of fundamental mechanisms. A child inherits one set of genes from each parent. If a trait is governed by simple Mendelian rules, the dominant version of a gene will reliably and predictably mask the recessive one.
Keyword being simple. Think a trait like the Achu syndrome. It's a genetic condition where sunlight causes a reflex sneeze, and it's a dominant trait. If you map a parent with the trait and a parent without it, you can calculate the exact probabilities. Pure math. The dominant trait executes its code.
Pure math, sure, if you only restrict your view to the absolute simplest traits in the genome. And that is where I have to push back. Mendelian inheritance is a neat introductory concept, but it fundamentally fails to describe the vast majority of biological reality.
How so?
Let's bring up the rule breakers, the complex traits that dominate actual biology. What happens when neither allele is dominant?
You're referring to incomplete dominance.
Yes, and we need to look at the why behind it, not just the what. If a plant with a red flower allele and a plant with a white flower allele reproduce, they don't produce a red flower like Mendel's rigid hierarchy would suggest. They produce a pink flower.
Right.
The strict order you just described collapses.
Well, it doesn't collapse. It simply introduces a different mechanism of expression. The alleles themselves are still predictably inherited. The pink flower just means the cellular machinery is reading the genetic recipe differently.
Exactly. Let's talk about that recipe. A gene for a red flower isn't just a conceptual tag, it is a literal recipe for manufacturing a red pigment protein.
Yes, it tells the ribosome what to build.
Right, and in complete dominance, one gene makes enough pigment to color the whole flower. But in incomplete dominance, the red gene only produces 50% of the required pigment, and the white gene produces none. The result is a physically diluted pink.
I follow you.
The expression is entirely fluid, and it gets more complex. Look at codominance.
Where both traits express at once.
Right. If a creature inherits a blue allele and a green allele for a skin pattern, it doesn't blend into teal. It produces a blue and green spotted pattern. Both alleles fight for expression and both win. And then we have multiple alleles interacting.
Like human blood types.
Precisely. Human blood types don't operate on a simple binary of dominant and recessive. We have A, B, and O alleles all interacting.
Which leads to A, B, AB, or O types.
Right. If a parent with an AO genotype and a parent with a BO genotype have a child, they can produce any of those. The combinations are vastly more intricate than Mendel's pea plants suggested.
That's an interesting point, though I would frame it differently. Even these rule breakers follow predictable mathematical patterns of inheritance once we understand their specific mechanisms. We can easily map the blood type probabilities on an expanded Punnett square.
You just need a bigger square.
Exactly. The complexity doesn't negate the underlying order. It just means the instruction manual has more chapters than we initially thought.
If you want to talk about chapters in the manual, let's talk about the pages that actively cross out other pages. I'm talking about regulatory genes. These are the ultimate agents of chaos in your ordered system.
I wouldn't call them chaos. They are highly specific.
But their entire purpose is to override, dictate, and completely silence the expression of other genes. Let's look at the Manx cat, famous for having no tail.
Right.
The gene for a normal tail is actually right there in the animal's DNA. The blueprint for the tail exists perfectly intact, but a dominant regulatory gene acts as a silencer.
Because it produces a protein that blocks the expression?
Yes, mechanistically, it produces a specific protein that physically travels over to the tail gene, binds to the DNA, and blocks the cellular machinery from ever reading the tail recipe. It actively turns the tail off.
I see that.
How can we call this a strictly predictable, ordered system when you have genes whose sole job is to spontaneously rewrite or suppress the expression of other genes? You could have the perfect Mendelian genotype for a trait, and a regulatory gene steps in and completely silences it.
I appreciate the mechanism of regulatory genes, but I must return to the concept of stability. A regulatory gene binding to DNA to silence a tail is still a genetic command being executed faithfully.
It's a very fluid system of checks and overrides, though.
But if the system were truly chaotic, the Manx cat wouldn't reliably pass down the tailless trait. But it does, because the regulatory gene itself follows the rules of inheritance. It physically copies and passes down through the generations.
It passes down, but the interactions it has with other genes are constantly shifting based on what the other parent provides.
But the foundation remains stable. Let's elevate this from the microscopic mechanisms to the macro scale. Let's look at how life survives long term. When we look at the survival of a species over millennia, the sheer scale of human success is a testament to the stability and reliable replication of our genetic code.
Do you think replication is why we succeed?
We are not succeeding because we are constantly mutating. We are succeeding because our DNA reliably preserves the complex traits that make us human.
Such as?
Our highly developed brains, our bipedalism, our internal organ structures. Every time a human child is born, it faithfully exhibits these traits, thanks to the strict rules of genetic inheritance. Mitosis ensures that once that child begins to grow, its brain cells develop consistently.
Okay, but you are looking at a snapshot of time and calling it permanent.
We conquer our environment because our biology provides a perfectly stable platform to build upon.
To understand biological success, you have to look at Darwin's theory of natural selection. Nature selects the most adaptable organisms, not necessarily the strongest and certainly not the most static.
But we adapt through our intelligence, which is a stable genetic trait.
We have that intelligence because of variation. This adaptability is entirely reliant on the constant, relentless variation generated by meiosis and complex genetic combinations. If our genetic code was perfectly stable and resistant to variation, we would still be single-celled organisms in a primordial soup.
That's a bit of a leap.
Not really. The only reason we have those highly developed brains and bipedalism is because our ancestors' genes varied, mutated, and broke the previous rules of their lineage, and looking forward, humanity is facing unprecedented environmental pressures.
Pressures that we are currently surviving.
Surviving for now. But look at the environmental realities outlined in the material. We're dealing with acid rain altering soil chemistry. We're dealing with a severe greenhouse effect driving rapid global warming and the depletion of the protective ozone layer.
And your argument is that we need random genetic variation to survive this?
I'm arguing that a rigid, mathematically perfect genetic code cannot pivot fast enough to survive a drastically altered food chain or a new biochemical threat. Only a deep reservoir of genetic variation will save a species.
I see.
When the environment changes, natural selection pulls out a new winning hand from the shuffled deck of alleles.
I am not convinced by that line of reasoning, because our massive population explosion, which is the undeniable metric of our biological success, isn't a result of sudden genetic variation. We haven't sprouted wings or developed radiation-proof skin in the last 200 years to deal with the ozone layer.
No, obviously we haven't.
Our population skyrocketed because our stable biological traits allowed us to master our environment. We used our predictably inherited cognitive abilities to develop agriculture, medicine, and technology. It is the uncompromising order of our biology that allows us to impose order on a chaotic world.
But the human brain itself is the ultimate product of genetic complexity. The cognitive abilities you champion are not governed by simple, strict Mendelian rules. They are polygenic.
Meaning they rely on multiple genes?
Exactly. Intelligence, behavior, and even our immune responses to new diseases are governed by dozens, sometimes hundreds of genes interacting in incredibly complex, unpredictable ways. They're heavily influenced by regulatory genes turning things on and off. You cannot point to the human brain as a triumph of simple, predictable order.
I'm not saying the brain is simple. I'm saying the mechanism that passes it down is reliable.
But rely exclusively on that strict replication, and we will hit a wall. Our static biological needs are outstripping the environment's capacity. If we rely only on the traits we currently have, perfectly replicated by mitosis, we are sitting ducks.
I think we have to recognize that the variation you are praising is severely constrained by the rules I am defending.
How so?
Because the mechanism of expression is universal. Yes, crossing over in meiosis shuffles the deck, but it's shuffling a deck of cards that is strictly defined by the DNA sequence. The organism is still bound by the fundamental laws of biochemistry.
Sure, transcription happens.
Right. The DNA must still be transcribed into an RNA messenger. That RNA messenger must still travel to a ribosome. And inside that ribosome, the genetic code is read 3 letters at a time, and specific amino acids are clicked together like beads on a string to build a protein.
The cellular factory.
That process, transcription and translation, is as mechanical, precise, and uncompromising as a physical assembly line. If that assembly line fails, if the RNA is misread by a single letter, the protein misfolds. No amount of grand genetic variation will save the organism if the baseline replication machinery is flawed.
I don't disagree that the machinery is essential. The assembly line has to work. But an assembly line that only ever produces one exact product will go bankrupt the second the market changes.
Unless that product is fundamentally necessary for survival.
But the environment always changes. You view the rules of heredity strictly as an assembly line. I view them more like an instruction manual that is constantly being edited.
A manual isn't meant to be poetry, though.
But life is biological poetry. The power of this system is in combining those genetic words in new, complex, sometimes rule-breaking ways to express entirely new traits. Incomplete dominance, codominance, regulatory genes physically blocking other genes. These take the basic vocabulary of the assembly line and create something uniquely suited to survive a new predator or a warmer climate.
But even poetry requires a strict adherence to the alphabet. If you randomly change the letters without rules, you don't get poetry, you get gibberish. And in biology, gibberish is fatal. That brings us to a crucial point of summary.
Fair enough.
My stance remains that life's foundation, its incredible durability across the eons, is built on the predictable mathematical rules of heredity and the flawless replication of mitosis. From the passing down of simple traits to the complex machinery of ribosomes building proteins, order is the primary currency of survival.
And my stance is that while order provides a baseline, life's resilience is driven by the constant variation of meiosis and the complex, rule-bending nature of genetic expression. Without the constant reshuffling of the genetic deck, life would be too rigid to survive a dynamic, dangerous universe. Complexity and adaptability are the true engines of biological success.
Well, we do find a point of convergence here, which is that DNA undeniably provides the ultimate instruction manual for life. Both the stable replication and the dynamic variation rely on the physical molecular reality of that double helix.
We certainly agree on the brilliance of the manual itself. The mechanics of how it is read are nothing short of miraculous. Where we continue to disagree is on how that manual is best utilized by a species, whether it is the strict adherence to the rules or the capacity to spontaneously rewrite the manual through variation.
Looking at genetics through both the lens of predictable order and the lens of adaptable chaos gives us a much more profound appreciation for biology. The study of genetics goes far deeper than basic traits or textbook diagrams.
Absolutely.
The deeper you look into the molecular mechanisms of life, the more you realize that every rule has a vital exception, and every exception relies on a fundamental rule to function. There is so much more to explore in the material to fully grasp these concepts.
Think back to that antique pocket watch we discussed at the start. Perhaps life isn't just the watch, rigid and perfect, but it also isn't just the boiling geyser, chaotic and formless. Perhaps the greatest miracle of genetics is that life is a pocket watch that can somehow invent its own new gears as the temperature rises. We leave you to form your own conclusion.